How's it going? My name is Ian Gomez. I'm in the Snyder Lab Department of Genetics, Stanford Cancer Institute, Stanford University School of Medicine, Department of Biological Sciences at California State, CSU East Bay. The title of my talk is Generative Immunotherapy, Ligand-Receptor Map of Immune Checkpoint Inhibitor in Precision Immuno-Oncology. And I wanted to go over the curation and overview of Lignature. Lignature is a curated database, encompasses intercellular transcriptomic signatures for 362 human ligands that are significantly expanding the repertoire of ligands with available intracellular response signatures. So they're ligand genes symbols with full names, aliases, and experiment keywords. So the metadata in the gene expression omnibus geo datasets are collected using GeoQuery, and there's an auto search. There's auto searching candidate geo datasets, manual curation geo datasets for each ligand gene, and data processing. As well as after the data processing, there's gene expression metadata, KEGG pathway database, as well as lastly the gene log fold change with pathway enrichment scores. Essentially, what this tool Lignature does is it takes bulk RNA and single cell RNA-seq data. So we can have two conditions in single cell RNA-seq as well as bulk seq and input the data for ligand signatures. So you can have L1, L2, all the way to LOK for each signature. And you get Lignature scores. So these will be like the L1, L2, all the way to LK for every SIG1, SIG2, SIG3, all the way from SIG-M and SIG-N. And what we get out of that is enriched pathways that are normalized, have a normalized enrichment score for each pathway. So the Lignature score versus the LR interaction score is a plot that we generate, as well as the selected Lignature score versus ligand expression and Lignature score. So we also get the average expression of ligands, conditions, cell types for all LR pairs, and the Lignature score, average expression receptors, and LR interactions and heat maps, as well as chord diagrams. And so overall, Lignature is able to calculate the LR interaction strength using cell phone DB, single cell signal arm, NAPME, and there are specific methods in the LR loop database initially. So Lignature compiles the signatures from published transcriptomic data sets and established resources such as CytoSig and Immune Dictionary, which are two extensive databases that are able to generate both gene and pathway-based signatures for each ligand. And applying Lignature to predictive active changes driving transcriptomic changes in controlled in vitro experiments and real-world single-cell sequencing datasets. Lignature outperformed exciting methods such as NicheNet, which was able to achieve accuracy in identifying active ligands and at both gene and pathway levels. So we have Lignature is able to establish a robust platform for ligand signaling inference, providing a powerful tool to explore ligand receptor interactions across diverse experimental and physiological contexts. We can see in a pie graph how many ligands there are from Lignature. So there's 213 ligands, and then from Immune Dictionary, there's 32, and CytoSig, there's 63. So together with CytoSig and Immune Dictionary, there's 54, and then 213 in Lignature. And when we look at all four databases, as well as the combination between the two for CytoSig and Immune Dictionary, we can see most of the ligands are in the CD molecules, cluster of differentiation molecules, and chemokines, as well as the growth hormone or IgCAM family. They're pretty close together here. Looks like it's IgCAM or growth hormone. There's a growth hormone family, and it's in blue. So the blue indicates the Lignature. as well as the CD molecules has mostly lignature. So all the CDs are from that database. And then also we see interleukins, which are the ILs like IL-6, IL-1, IL-2, all the interleukins, which mostly are in cytosig and immune dictionary. There's also neuropeptides, mostly lignature, and TNF-SF, which is a tumor necrosis factor, soluble factor, which has both cytosig and immune dictionary as well as immune dictionary. Next, we have the LRI predictions in melanoma, which is a skin cancer and the myeloid lineage. So all the innate immune cells that are myeloid in the context of colitis, inflammation of the colonocytes, and normal colons in C. And so they have immune checkpoint blockade associated immunotherapy. And we see a SLRI score that's classified with both high, both low L score and a low SLRI, lignature score, and the differential expression of ligands in the myeloid colitis and NC cell type LR pairs. So you can see ligand similarity scores predicted by lignature and the maximum LRI strength scores calculated by the method NAPME across all sender cells and conditions for all the LR pairs with the scores available in the data sets. So the normal colons are CI treated, cancer immunotherapy treated melanoma patients who underwent endoscopic evaluation for suspected CI induced colitis, but were found to have normal colonic mucosa endoscopically and histologically normal colons. And we can see mostly the myeloid cells in the colon, in the colitis and normal colon, and then that have higher levels, and mostly CXCL9, SERPINA1, LIRB4, interleukin RN, and let's see, a TIMP1, TGN2, TNF, SF13B, and ITGAV, which is an integrin. MIF is in a cyclin T normal colon, cyclin T colitis. We have HMG and CD4 T resident memory cells in NC. There's let's see, CD40 LG, and let's see, another TNF SF9 in a CD4 tissue resident memory cell, RMC and NC. So essentially, these are the similarity scores predicted by lignature and the maximum LRI strength scores calculated by the method NAPME. They have both sender cells and as well as the conditions for all the LR pairs that are available in this data set. So this shows ligand receptor interactions with immune checkpoint blockade associated immunotherapy for melanoma. And then here we see the differential expression. These are the same one, differential expression, just a little bit larger. And then here we have the enrichment pathways that are normalized in myeloid colitis versus the normal colon. We can see central carbon metabolism in cancer, natural killer cell mediated cytotoxicities, NK cell mediated cytotoxicity. So the NK cells, human immunodeficiency virus 1 infection, glycolysis, gluconeogenesis, NOD1-like receptor signaling pathways, RIG-I-like receptor signaling pathway, JAK-STAT signaling pathway, EB virus infection, Epstein-Barr virus, insulin signaling, and PD-L1 expression, PD-1's checkpoint pathway in cancer, which is a common immunotherapy. There's also focal adhesion, hedgehog signaling pathway in glycan biosynthesis and dilated cardiomyopathy. So these are all the enriched up and down regulated KEGG pathways, myeloid cells between colitis and with CI cancer immunotherapy and normal colon NC. Next to this, I have the ligand expression and lignature score together for the ligand max expression, where you can see the specific cell type as well as condition. And we can see B2N is a beta 2 microglobulin, which is on the MHC molecule, major histocompatibility complex, and the TCR, the T cell receptor. There's also S100A8, HMGB, LTB, LGALS1, CD2, TNF, and the highest is interferon gamma, which is an exerkine molecule. Now we can see that a little further to the right in pink, and the cell type is a naive B cell. And let's see, ANXA1, ANXA2, CSF1, SERP BP, and CSF2. There's a cluster together with the CD40 LG LIF, leukemia inhibitory factor, LIF, TGF beta 1, OSM, which is similar to IL-6 family. And let's see, CAMP, TNF, SF12, and GAS6 and TNF, SF13. So these are the lignature score versus ligand expression max, where we can see the similarity scores predicted in the maximum average expression levels across sender cells and the conditions of ligands identified LRIs with the detection rate of the ligand genes above 0.25 and any cell type detection rate genes above 0.25 and the myeloid and ligand scores that are above 0.1. So I wanted to show you the average of the ligands, the heat map where we see exercise immunotherapy LRIs. So specific LRIs that are involved in exercise. So these are interferon gamma receptor 2, interferon gamma receptor 1, LIF, IL-6 signaling transducer. So this ST and IL-6 is soluble. It's more cytoplasm than the membrane. IL-6 by itself is on the membrane, diffuse once it binds to IL-6 receptor. But this ST is most likely in the cytoplasm, which is subcellular, intracellular. TNF and TNF-R SF1B, TNF-FR, TNF-RS F1A, OSM, IL-6ST, TGF beta 1, ITGAV, TGF beta 1, and TGF beta receptor 2, 1 and 2. And we can see the different cell types. So starting here with the CD4 T cells, we have resident memory cells, CD4 T regulatory T cells, and with follicular cells. So two different T cells, regulatory and follicular helper cells, which is kind of like a unique population for T cells. Usually it's T helper cells and T killer cells, but these two are CD4 T regulatory and T follicular helper cells. Naive B cells and are there as well. We can see the LRIs for those over here in the heat map. And cycling T mast cells, myeloid cells, and ILC2 and ILC3 for each cell type as well as condition shown here in this heat map. So we can see the average expression here in the context of immune checkpoint associated immunotherapy. Here's a similar pop only on the myeloid cells. So just the myeloid cells, we can see LGALS1, PT, PRC, and CD2, CD48, which is a novel interaction, and TNFS, F13, TNFR, SF14, or excuse me, the beta 2 microglobulin, LILRB, little rb1 in the myeloid C, and then CSF2, CSF1R in the myeloid NC, and the CSF1, CSF1R in the myeloid NC2. So we have both conditions, myeloid cell, all the LRI's predictions for melanoma or melanoma in the myeloid cell for C and NC ICB associated immunotherapy with Lignature scores that are absolute myeloid cells in colitis and myeloid cells in non-conditioned samples. So here we have the specific L score, where we can see the higher expression on the top, interferon gamma receptor 2 and 1 with interferon gamma, and the TNFR, SF1A and 1B, as well as LTBR and TNFR, SF21. Other significant interactions are LIF, IL-6, ST, Gas6, MERTK, receptor tyrosine kinase, Gas6 and AXL, AXL, and B2M little 1, B HLAF, and XA2 TLR2, which is a toll-like receptor, and ANXA2 is another interaction that we found in the precancer atlas. And then there's CSF2 and CSF2 receptor, and Serpina LRP. So we see Lignature scores, which is the L scores here, that are candidate ligands that are based on the user's choice for a similarity measurement metric. Where we have the Pearson correlation coefficient, Spearman correlation coefficient, cosine similarities, and the Euclidean distance, where we have the signature weights from combinatorial analysis by regularization linear regression that has a ridge, a lasso, or an elastic net. And I was able to choose specific to exercise immunotherapy, LRIs. So lastly, I wanted to show a Circos plot of receivers and sender cells with the LRIs. And we can see the different cell types as well as ligands and receptors. So the receivers are the ones with the arrowheads, and then the senders are with the blunt ends. And so we have myeloid cells in yellow, and then the memory Bs in this light blue, and purple we have CD8 cytotoxic lymphocytes, CTLs, and then their ligands like interferon gamma, TNF, and TGF beta 1 with CD8s. Light yellow we have CD4 tissue resident memory cells, TNF, CD40 ligand, LG, and TGF beta 1 in that specific cell type. And then in dark green, we have CD8, LP, interferon gamma, TNF, CD2, TGF beta, and then we have CD4, T regulatory and T follicular cells, that interesting population between regulatory T cells and follicular helper T cells. And then naive in blue, and in blue we have naive B cells with BTLA, and TGF beta 1 and LTB. And then in red, we have IG plasma B cells with MDK. And in brown or light brown, we have cyclin T, HMGB, LGALs. In a lighter green, we have mast cells with TGF beta 1, LGALs 1, mast LIF, and group ILCs, which are lymphocytes, GCs, myeloid, ILCs, myeloid, LTBR, CD40, and so these are on the bottom we have the blunt ends, and on those arrow end, and then each interaction, like these pink ones, ILC2s and ILC3s, we have CSF, TNF, LTB that are going to the myeloid cell with TNFR, SF1, and then this one goes as ILC2, ILC3, CSF2 goes to the CSF2 receptor beta, receptor alpha, 1 receptor, CSF1 receptor, and CSF3 receptor on the myeloid cells. So we can see that the interaction happening with multiple cell types as well as ligand receptors. And yeah, it's a good example of how Circos plots work with sender receiver cells and ligand receptor interactions. So that's just essentially Live Nature, how it works for LRI predictions. And here we have melanoma, the myeloid lineage, colitis, and normal colons that underwent immunotherapy-associated immune checkpoint blockade. I want to thank you for your time. If you want to send me any questions, feel free to reach out to me. Thank you.